Awesome. Well, it's great to be here today in Austin, Texas. I appreciate you all coming out. I realize there are a lot of things that you could be doing on a Saturday morning besides listening to a neuroscience lecture, but I hope that you will enjoy this, and I can already sense that folks here are engaged in learning science, engineering, and other related fields, so I think we're going to have a great time. I imagine you will have a lot of questions, and that's great with me. And please take note of those, and I'll be sure to answer those at the end. I've also got my email on a final slide, so you can send me a note if you'd like to discuss further. So thank you again. So I want to go over kind of a brief agenda for my talk today. First, I'll give a disclosure. I want to share my testimony as a Christian. This is an important part of a presentation in this context. Some background on the topic that I'll be discussing today, our major objective, our methods and materials that were used in this research, results, discussion, conclusions, and then time for questions. So in terms of the disclosure, I want to share that the views and opinions that I will give today are my own and do not necessarily represent my academic affiliations or my current or former employers. So in terms of my testimony, I am from Western North Carolina, where the hurricane hit recently, and we certainly appreciate your prayers for our region and the folks there. My training is in neuroscience, so I have a PhD in neuroscience and a bachelor's degree in biology, but I consider myself to be multidisciplinary. As John mentioned, I'm interested in archaeology, but I also work heavily in other scientific disciplines like health economics and outcomes research, comparative effectiveness research, and currently my role is in consulting with large pharmaceutical companies bringing new drugs to the market or helping them understand and create evidence packets around existing therapies as well. So I love science, and that for me is guided by my love for the Creator. My mother, who is here today, Cheryl, led me to the Lord at the age of 6, and my love for science stems from my love for the Creator. So that I want to make that very clear. Early on, you may agree or disagree, and that's great with me. Let's talk about it more later, but that's my testimony and the story that I'll be sharing, because I believe the best science is shared in stories. Is some of my favorite research that I've been a part of in my scientific career. So our understanding of neuronal types and circuitry has expanded drastically over the last 100 years when Ramon Cajal pioneered the field of neuroscience. And if you follow the news or if you're into more academic settings, what happens in the peer-reviewed literature, you'll know that there continues to be just a tremendous amount of evidence that is being generated over time. However, it's been long recognized that there is a need to establish a computational framework that may underlie the brain's functions. So I want to ask you to ask yourself this. Imagine if all the components were available to you, what mathematical principles might have been used in constructing brains that are capable not only of cognition, but also adaptive behavior? So keep that question in the back of your mind as we move through the background. Clearly, this is a very complicated question, and there are many ways to approach it, and perhaps several different answers. The human brain is very intricate. It is estimated to have around 86 billion neurons, and each neuron has tens of thousands of connections. So that means that there are over 100 trillion synaptic connections that exist within each of our brains. And if you're like me, sometimes you feel that you have far fewer than those, and they don't always come together at the right times. But that does exist within each of us that are present today. Another layer of complexity is that not only do we have all of these cells, but there are different types of cells, and their connections with one another change over the course of our development and as we age. Within humans, but across species as well. So as such, the underlying mathematical computation that enables all of this to take place has been considered by many neuroscientists to be the utmost difficult question in our field. And I'm going to try to answer that today through some research. So recently, the lab that I was a part of in graduate school took a different approach to answer this question in what we consider to be a thought experiment. So we speculated that the essence of intelligence lies in our brain's ability to uncover specific features, but also generalized knowledge. This is coming from a world of uncertainties and infinite possibilities, and I feel that we can all relate very well to that. So how can a brain be capable of understanding, perceiving all of these events that can take place, but to generalize concepts out of those? So based on this, we surmise that our search for the brain's logic can be reduced to a question of how neurons are connected in such a way that would allow the brain to uncover various patterns and generate conceptual knowledge. So as you can imagine, there is a long list of questions that one might ask. The first is, how do excitatory neurons within a cell assembly organize themselves? And I'm highly aware that some of the phrasing that I will be using today is field specific, and I'm going to try to explain these as I go. And if there are areas in which you have questions at the end, hey Grace, what is an excitatory cell? I didn't cover that. We'll talk about that at the end. So very much aware of that, and I hope I can kind of make that clear as we go along. Excitatory cells are one type of cell within the brain. They are predominant in the forebrain, and in recent years have been very much of interest to scientists. This is in contrast to other types of neurons. Perhaps you've heard of dopaminergic neurons or serotonin. Those are neuromodulatory neurons. There are also inhibitory neurons. What we're focusing on are the major types of cells in the forebrain. A cell assembly is just a group of cells. If you are all neurons, then this group of folks is a cell assembly. So how should these be organized? And from this organization of cell assemblies, what enables us to separate patterns and to generalize patterns? How do we get at conceptual knowledge? In an abstract sense, and I realize up until this point it's all been quite abstract, is there a mathematical principle that is conserved across neural circuits that is intrinsic despite their anatomical features? When you look at a brain, it appears to be quite homogeneous from the outside, but when you dig into the details, you realize that there are many different brain structures. They have different types of neurons, and their connections are different between regions, and how they're connected to other regions within the brain is variable as well. So the question is there a principle that is repeated throughout the brain regardless of the structure in which that's taking place? A great question for the field is whether this logic is dependent upon learning in adulthood or pre-configured by genetics. So the field of neuroscience the subfield that I've been most heavily involved in is learning and memory. So maybe you've heard of Eric and Dell, for example, cells that fire together, wire together. The question is, if there is a mathematical principle that's available, is this pre-configured by design, or does it appear and change over time? And finally, if there is such a computational logic, is it implemented throughout the cortex? And if so, to what degree? And does this allow for greater intelligence? As we all know that the size of brains differ from species to species, but the degree of intelligence perhaps it's not always correlated with the size of the brain. And I'll let you think about different animals that you know and whether you feel that you're more intelligent than them, even though they have a larger brain. I won't answer that for you, but that's something for you to consider in your own time. So what I want to describe is a theory that our lab put together, and we want to explore the questions that I've just posed with some basic principles. This is a bit text heavy, I realize that, but I think for some folks it's helpful to read as I speak, and then I will show what I consider to be some cute diagrams for the rest of us that like to see things quite visually. So this theory specifies that within each computational building block, which we're calling here a functional connectivity motif. This is a set of neurons. The total number of excitatory neurons, so these are that group of neurons that we discussed previously, with distinct inputs should follow a power of 2 based equation. So n equals 2 to I minus 1. N is the number of neural cliques that can cover all possible permutations and combinations, and I is the distinct number of information inputs. Let me explain that in another way. Each FCM or group of neurons contains excitatory cells that receive specific input as well as those that receive progressively more convergent inputs. This allows these neurons to capture and encode every possibility that they encounter. So this arrangement allows cell assemblies to code very specific inputs, some that are subcombinatorial, so input 1 and 2, or input 2 and 3. And finally, general responses to inputs 1, 2, 3, and 4. So here is an example of that. Each of these kind of triangles is a different neuron. I here are the different inputs. So you can see that when there is one input, cells may have one individual response. They may each respond to that specific input. When there are 2 inputs, a cell may respond to input 1 and 2, 1, 3, 1, 4, 2, 3, etc. And then finally, if there are 4 distinct inputs, there are cells that will encode all 4 of those inputs. So we're speculating that the neurons, or the cliques, these are assemblies or groups of neurons, like we are here gathered as a clique or an assembly of people. These unique specific cliques, they extract very unique information about the environment, perhaps external or internal sources. And we are hypothesizing that subgeneral and general neural cliques categorically extract all combinations. And remember, we're thinking about a brain that has 100 trillion connections. How can we make sense of that? How can these cells encode every possibility that we will encounter throughout our lives? So again, these specific or general cliques or groups of neurons encode specific features, maybe a memory or an action. General cliques allow for conceptualization, so semantic memories, categorical knowledge, general intent, and motor instruction. So to summarize, this logic, if it's true, would allow each group of cells within an assembly to encode every possibility that they may encounter in a specific to general manner. So how can we test this framework? And if I've already lost you, I'm going to find you again. Don't worry. So the brain is designed such that there is a structure of function relationship, meaning that we may be able to detect these patterns in cells of living animals and try to uncover whether there are specific to general coding features. So moving along here, this is called hierarchical clustering plot. N here refers to numbers of neural cliques. This orange represents one set of neurons or cells that respond to one feature. They all activated. So in this case, activity is orange, blue is the absence of activity. So neuronal clique excitatory click 1 responded to pancakes, number 2 responded to milk, 3 to eggs, and then 4 to blueberries. As we go down, these become subcombinatorial. They're responding to pancakes and milk, perhaps milk or pancakes and eggs, pancakes and blueberries. So these combinations increase until we get to groups of neurons that encode all inputs, pancakes, milk, eggs, and blueberries, and this is a breakfast meal. So you can see that from these specific inputs, we're conceptualizing these and getting to the concept of a breakfast meal. So the theory predicts that by providing 4 distinct inputs, so in this case I equals 4, we should observe 15 neural clicks in relevant brain regions that exhibit these properties. So what I'm saying is that if we can record the activity of cells in relevant brain regions and provide those cells and thereby those animals with 4 inputs, we should be able to record and find these coding patterns. So this theory has several testable predictions, and I want to walk through these, and we're going to cover some of these today and then leave some for a later time. So the first prediction is that this logic should be used to process all types of cognitions that we would encounter in our lifetime across modalities, so appetitive, social, and emotional information. This connectivity logic should be prevalent across many cortical and subcortical regions, regardless of the variations in those. And I described that there are many brain structures, they vary in a lot of ways. What we're looking for here is something that resides over those as a basic mechanism by which these cells encode experiences. So we've talked about excitatory cells. I said those are the major cells of the forebrain. They're involved in a lot of learning and memory activities. There are other types of cells. I mentioned dopaminergic cells. We suspect that they use a different logic, and I'll describe that a bit later in the presentation. We are suggesting that this organization is developmentally pre-configured and does not depend upon learning in childhood or adulthood, so that this resides over the learning and memory experiences that we all have. This is by design and is innate. Something that I won't get to in this presentation, it could be its own presentation, is that we have some ideas for why and how this logic may be repeated across different parts of the cortex. That we all have large cortices. The surface area of those differs from animal to animal. Is there a residing principle behind those? And we think that there is. The last point I'll make is that we are suspecting that this logic is conserved across the brains of different species. So not only humans, but also different types of animals, and we'll be showing the results of those testings today. So as I said, we will focus on, for sake of time, those that are highlighted in bold here, and then if you have questions about the cortex and how this logic holds true and is implemented across different cortical layers, let's talk about that later, and I can also share the manuscript with you so you can read it in your own time. So the objective was to test these predictions and see if they hold true. And to do that, we employed some very sophisticated recording techniques. So these are large-scale in vivo recording methods to measure the activities of cells in freely behaving animals. So this was done in a laboratory setting. We used hamsters, rats, and mice. Basically, you build an electrode, and I will show you some examples of that, and implant it in a brain region of interest, isolate the cell types of interest, and see if those activity patterns hold true when you give a specific number of inputs. So if you've not seen one of these before, this is a tetrode recording system. Basically, you make each of these by hand, and they are crafted fit for purpose for the brain region of interest for you. If you're interested in the hippocampal CA1 area, your your setup is quite different than somebody that's interested in recording deep in the brain, perhaps dopaminergic neurons. So these are made, this is a coronal section of a mouse brain, so you can see that we can implant deep in the entorhinal cortex here, in the hippocampus, midbrain, and then throughout the cortex as well. So this is done in freely behaving animals. You definitely want to check that you've implanted your electrodes in the right spot. So after the recording experiments are complete, you sacrifice the animal and slice its brain. Again, this is a coronal section of a mouse brain, and you can use dye to trace where those electrodes have been. And that's important because if we've implanted in the wrong place and assigned those activity patterns to an incorrect brain region, then the results are not very meaningful without the context of the location. So what does this look like? I realize if you've not seen this before, it looks a little funny, and we did swap out the smiley face balloons for Christmas balloons when the time of year was appropriate. But basically, we would implant these recording systems into the brain of a mouse, and they walk around with these attached, and there's a balloon filled with helium to offset the weight of the recording system. So this is recording their brain activity in real time. So this plot, it may not look like much, but this validates that these animals are freely behaving. These tracings show the activity patterns of this animal over a given period of time, and that they're able to live and move as they normally would. So these are plots that help us separate different types of neurons. So these red groups of cells that you see here are recordings of different individual neurons, and we are separating them from other types. You can see that the waveform, and this is what happens when there is an action potential or when a cell fires. For an excitatory cell, the peak and the width of the waveform is quite different than an inhibitory interneuron. They typically have these very steep waveforms. So we were able to separate those using different methodologies so that we can exactly say these are excitatory cells from the hippocampus, not inhibitory interneurons. And if you're interested in those and how they may fit into this, let's talk later. It's just a little bit too much for today. This is local field potential. We are interested in the activity of individual neurons, and that is a highlight of this technique, that we can, we're not only looking at the activity of many neurons, but those individually. However, this shows that there is stability of the recording system over time. If for some reason the apparatus came loose, or there was noise interfering, this local field potential would be heterogeneous, it would be unstable, and that would be apparent to you. So this is another sense check to us to say, hey, you implanted an electrode, it is stable over time, it is fair to use the data that have been recording from this animal. So you have implanted an electrode. The mouse or the other animal is doing fine. You have neurons that you're recording from. What does that data look like? If you've never seen one of these before, these are peri-event rasters. If we take a look at the top left, 0 is when an event takes place. The event type doesn't matter here for the purpose of this illustration. You can see that the firing of this neuron increased after the event took place. And these lines, which are quite small, appeared 7 times. So this is 7 times when that event took place. This is the average response over time. These that are grayed out show that this neuron had no response to 0 when that event took place. So when I say throughout this presentation these cells responded, what I mean is that there was a statistically significant increase in their firing when the event took place. So we can transform this type of of graph into what I showed you before. This is a hierarchical clustering plot. Again, orange here indicates that these cells responded to that input. So you can imagine that all of these that increase their firings would belong in this orange spot. Those that did not have no response, and those are blue. So when I say that there are activity patterns, this is what I'm talking about. Did these cells respond? Did they respond to all of the stimuli or just some of them? That's what we're going to tease apart here. So to check whether this logic is implemented throughout the brain, first we went to the basolateral amygdala, which you may know primarily being a part of fear circuitry. Perhaps we've all heard of the almond-shaped amygdala. It's also involved in a lot of other activities, and including food circuitry. So we gave mice 4 different types of food. Note that cheese, I don't know why cheese was not on the list, but they do like all of these, their regular rodent diet, their sugar pellets, rice, and milk droplets. And then to avoid training-induced alterations in their firing, we use data sets from the very first time that they consumed these. So the first 7 times they consumed milk, first 7 times they consumed a rice grain, and this allowed us to prevent any changes that would happen through learning these events over time. So these are individual units. A unit here is a synonym for cell. In this case, these are excitatory cells. We have the rodent diet, sugar, rice, and milk. On the left, you see hertz. This is the number of firings per second. So you can see that the first neuron, or unit here, A, only responded to milk. The second responded to rodent diet and milk. The third responded to 3 of the events, and then finally this general unit, what I mean by general is that it responded to all 4 of the stimuli. So we found what we consider to be event response selectivity, where some cells responded only to 1 type of food, others responded to 2 or 3 types of food, and then others responded to all 4 types of food. Let me show you what that looks like when you transform it into a hierarchical clustering map. This is a group of cells that responded to rodent diet only. The changes in color here, as you can see, 0 to 1.2, it indicates the magnitude of the firing. So you can see that all the cells increased their firing that were above blue. Here are sets of neurons that responded to the rodent diet and sugar, and then we continue on to the rodent diet, sugar, rice, and milk. You can see on the right-hand side we have the numbers 1 through 15. If you remember the equation that I gave, when we have 4 specific inputs, we would imagine that there would be 15 groups of neurons that would respond to those, and that's what we found, 15 different neural cliques that encode not only specific inputs, rodent diet or milk, they respond to those subcombinatorially, meaning that they respond to 2 or 3, and then finally there are a group of neurons that respond broadly to all inputs. What I'm showing here on the right in C is the same hierarchical clustering evidence from 1 rodent. So often you can see that the number of neurons changed. These are from multiple animals. These 50 neurons are from 1 animal, showing that of course we can, if the sample size is large enough across multiple animals, we can find that these coding patterns exist, but we can also find them within the brain of 1 individual mouse. It just depends on sample size. So just to summarize, as I was increased from 1 to 4, the plot showed the emergence of distinct neural cliques from 1, 3, 7 to 15. So our conclusion from this experiment is that basolateral amygdala excitatory neurons followed this specific to general logic for capturing various food experiences. So next we move to a different type of inputs. So we had just talked about appetitive experiences. Now we're going through an experiment where we're focusing on social recognition in the medial amygdala. So here there is a mouse that has a recording electrode implanted, and it has interactions with female mice and with male mice, either the face or the genital region. This is a very common way for mice to interact with one another, and we will be looking at the firings of the neurons at each of those intersections when they come into contact with the face of a female or a male, and all of the combinations in between. So again, you can see that the input type is listed on the top: female face, female genital, male face and genital regions. We found that there were neurons that responded very specifically to one of those interactions. There were also neurons that responded to 2 or 3 of those interactions, and then finally neurons that responded quite generally to all of those interactions. So again, what this demonstrates to us is a specific to general responsive pattern. So I've again transformed those individual data into a hierarchical clustering plot, and you can see that amongst these 235 excitatory neurons, there were 15 different neural cliques or assemblies that are encoding not only the specific features, the female face, the male genital, they are encoding every combination of those activities, and also broadly, there are some groups of cells that respond to every input. So this is further evidence that this logic holds true in a different type of experience. So next we wanted to move into kind of a final set of experiments that are focused on emotional activities. If you're familiar with the literature in the field of learning and memory, a lot of it has to do with fear, and how that is encoded and processed and stored in the brain. So for us in this area, it was kind of a classic road to go down. Your question may be, Grace, how do you scare mice? They normally scare me. I'm right there with you, totally right there with you. But we tried to mimic experiences that mice would have in their day-to-day lives had they not been laboratory animals. So we used an air puff blast, a free fall, an earthquake-like shake, and a mild electric foot shock. An air puff blast may indicate to them that there is an owl or another animal that's flying above them. Falling is something that is native to all animals, I think. An earthquake-like shake, and then a classical piece of conditioning evidence is a mild electric foot shock. None of these injure the animal. I would describe them as being startling, but they do increase freezing behaviors and heart rate variability, and our lab has shown that over time. So they're scary, but they are not they're not harmed in any way. So this is recordings from the anterior cingulate cortex, so you could think of it as an area on the top or the outer part of the brain. We have 4 different units or excitatory cells, and they are responding to the air puff, the free fall, the earthquake, or the foot shock. You can see again that there are units that respond only to the foot shock, those that respond only to the air puff and the free fall, the earthquake, and the foot shock, and then some that respond to all 4 of the stimuli. So here I'm showing data that are transformed. These are 406 units from multiple mice and 76 units from an individual mouse recorded in the ACC. So you can see that there are 15 different neural cliques encoding these 4 different inputs, not only in data that are summed across multiple animals, but also in the brains of an individual mouse as well. So the next question is whether this prediction holds true across different brain areas. So I showed you some data in the basolateral amygdala, some data in the medial amygdala, some data in the anterior cingulate cortex. Does this hold true in other brain regions? So we perform the same experiments focusing on neural circuitry that is known to be associated with fear and memory. That is the infralimbic cortex, the retrosplenial cortex, where I did a great amount of work in the hippocampal CA1 region. So we'll, for sake of time, kind of skip the peri-event rasters that show the responses, example responses from neurons, but I'm showing here that for four distinct inputs in the infralimbic cortex, there are 15 different neural clicks that respond to the air puff, free fall, earthquake, and foot shock. So this logic is not only true in the regions that I just described, we're now moving on to different brain regions using the same inputs, and that logic holds true as well. This is the data from the retrosplenial cortex. You can see that there are n equals 15 different neural clicks that are encoding every combination of the inputs that we're giving them. Those that respond specifically to one input, those that respond in a subcombinatorial way, and those that respond generally. And just as a reminder, these at the very bottom, where there are colors that are not dark blue, indicate those that respond quite broadly to the events. Perhaps if you have a favorite brain region, for most people it's the hippocampus, and I understand why. A lot of work has been done here, and we are repeating here exercises in the hippocampus that we showed very early on in our lab before publishing on this theory. You can see here again with these four distinct inputs that there are 15 groups of neurons that respond to these. So not only is this true in the infralimbic cortex, it's true in the retrosplenial cortex, and true in the hippocampal CA1 region as well. And all of these have been previously implicated in fear memory. So this is exciting, and this shows that that four different regions have the same coding patterns, that these predictions hold true, that they're used repeatedly along the cortex and also in regions that project and receive projections from there as well. So I want to contrast this work focused on excitatory cells with the dopaminergic system, which is neuromodulatory in nature. We predict that because these neurons have different functional purposes, for example, they provide motivational signals like wanting or not wanting, rewarding versus not rewarding, that they would have a much simpler logic. So we investigated how dopamine neurons deep within in the brain, the ventral tegmental area, encode emotionally fearful experiences. I want to show a waveform for a dopamine neuron. You can see how different this red line is compared to those excitatory cells. This is a way for us to demonstrate to ourselves and to others in the field that yes, these are not excitatory neurons that we've been recording from previously. These are dopamine neurons located deep within the brain. There are many other ways to test whether this and to verify whether these are dopamine neurons. If you have questions about that, happy to answer that at a later time. So we found that there are three major types of dopamine neurons. The first type, type 1, suppresses their activity. Initially, at 0 here is when the event takes place. They initially suppress their activity, and then they have some rebound activity. You can see that there's kind of a blank space here for a little over half a second, and then there's rebound activity. We found that there is a separate type of dopamine neurons that initially suppress their activity in response to the event, but they do not show the substantially different rebound excitation at the end. And fourth, there is, or third rather, there is a group of dopamine neurons that increase their firing to all of the stimuli. What's interesting about this is that there was no specific tuning of these cells to the stimuli. You know, when we were just going through the results for the excitatory cells, we found that there are cells that respond to one input, 2 or 3, these cells are broadly tuned and they respond to all of the events in a similar way. You can see that I think about 82% responded to 4 fearful events, and the remaining responded to 3 events. So there were no neurons that dopamine neurons that responded specifically to 1 or 2 events. So this suggests to us that dopamine neurons lack the coding pattern that we found in excitatory cells throughout the brain, and that their logic is different, and that they are broadly tuned to fearful stimuli. I want to quickly go over what I think is the most exciting part of this research, which is, some time ago, Hebb postulated that cells form assemblies through the act of learning. I mentioned cells that fire together, wire together, and perhaps this is what organizes cell assemblies. Our theory predicts, however, that the logic that we've described and demonstrated throughout the brain is pre-designed or pre-wired and is not dependent upon learning. So you may ask, how do you test these hypotheses? So we deleted NMDA receptors in the 4 brain excitatory neurons. The NMDA receptor you may have heard of is kind of the synaptic gate for learning and memory. Without an NMDA receptor, animals are unable to learn and remember things. My graduate school mentor created the NMDA receptor knockouts that we used here. So something that he's been excited about for a long time, and they came into play very, very well for this set of experiments. So the question is, we've tested wild-type mice. Now we're going to test those that lack the ability to learn and remember. Can we still find these coding patterns in the brain? So this is data from the basolateral amygdala again. Wild-type mice, WT, and knockout mice, KO. You can see that between these 2 hierarchical clustering plots, the evidence is largely the same, if almost identical, indicating that there are 15 neural cliques that respond to these 4 stimuli, regardless of whether learning, these animals were capable of learning and remembering. So those are repetitive data from the basolateral amygdala. And next, I'll move to the retrosplenial cortex again and the anterior cingulate cortex. Here we're investigating fearful experiences. You can see that between the wild types and the knockouts for both brain regions, there were 15 neural cliques that responded to each of the stimuli in a specific to general manner. So whether these animals could learn in adulthood, these activity patterns were evident, and we were able to measure them. So this demonstrates that although you delete NMDA receptors, which are essential for learning and memory, we can still identify this specific to general logic in the brains of mice in 3 different areas, 2 in the cortex and 1 in the amygdala. So this is, I think, the most exciting part of this research. Finally, and quickly, we wanted to test whether this logic holds true across species. So the data that I've shown up until this point were in freely behaving mice. Next, we moved to Syrian hamsters in the prelimbic cortex, again subjecting them to fearful events. These are data from a hamster. You can see very, very similar to that of a mouse. We have specific units that respond to 1 of the inputs, those that respond to 2 or 3, and then finally those that respond generally to all 4 of the inputs. And again, we found event response selectivity that we found in mice. So I'm showing a transformed plot indicating that amongst these 125 neurons, we found 15 different groups of neurons that encoded these 4 fearful experiences in a specific to general manner, covering all possible combinations. So that's the recap on the results section. Let me just summarize quickly, and then I know you have questions, and I'm happy to take those, as I mentioned. Here we systematically tested many of the predictions from the theory of connectivity. And we found that this power of two based logic is widely implemented in the brain in many cortical regions and subcortical regions and across two different animal species in experiencing appetitive, emotional, and social events. These findings are important for several reasons, and I'll outline a few here. First, this principle is quite simple, but it allows cells to deal with every possibility that they can encounter over a given period of time by generating every possible connectivity pattern. This logic fulfills an essential requirement as a unifying principle because it can cover neural networks that are very simple and also those that are very complex. It fulfills the need for this to be, again, unifying in that we saw these activity patterns in multiple cortical regions, and again supports the notion that this is implemented widely across the brain, specifically in excitatory cells. We noted that dopaminergic cells are a great contrast to this, where their tuning is quite broad compared to these excitatory cells where they are tuned in a specific to general manner. As I said, excitingly, we found that when we removed the NMDA receptor, there was very minimal impacts on the coding patterns, suggesting that this wiring logic is pre-wired by design and independent of learning in adulthood. So I want to summarize quickly. So I believe that these data do suggest that the theory of connectivity is used widely across the brain, and that it governs how cell assemblies or groups of cells are organized so that they can encode specific to general information, giving rise to conceptualization and abstract knowledge. It is imperative to continue to test whether this theory of connectivity is implemented in other species. There are many different types of neurons, and they modulate the activity of excitatory cells. You can imagine that these patterns may vary in disease models, so in models of Alzheimer's disease, in models of schizophrenia. So understanding how these activity patterns change or do not change will be very informative to those diseases. This, for some folks, that might find this quite interesting. Neuroscience broadly as a field has been moving in the direction of artificial intelligence for some time, and these findings can support those efforts, I think, in a tremendous way. So I'll pause there for questions, and my email is here if you don't get a chance to talk to me at the end. So thank you all for your time, and looking forward to the questions.
All right, thank you very much. Appreciate that. I'm so thankful we had your presentation before the presentation on marijuana. I don't think I have enough brain cells after that one to be able to digest this. So the Q&A, two rules real quick. So first of all, just to let you know, we do have two microphones. David has a microphone. I also have a microphone. If you do have a question, raise your hand. We'll bring you the microphone. So we want you to ask your questions in the microphone. And we do want you to ask questions. So sometimes in the Q&A, somebody will take over, and then they'll go on a long rant for 10 minutes. We're all waiting for that question, and it never happens. So, don't be that person, please. If you need to set up your question with some commentary, then please do so. But otherwise, please ask a question so we get to other people. And the second rule is, I'm the president. I get to ask the first question.
Okay.
All right. So I think this is a difficult question. But as a Christian neuroscientist, I'm extremely interested in the answer. So neuroscience is mostly a deterministic field. From a philosophical point of view, they study it from a deterministic perspective. My question is, how do you, as a Christian neuroscientist, understand or imagine how the soul interacts with the brain? And just a quick example, Alzheimer's. With Alzheimer's, we're learning that the plaque in the brain causes it to lose function. And my grandmother had Alzheimer's, and she declined to the point where she was no longer there. she was unable to express herself outwardly. And so that's always made me wonder, how does the soul interact with the brain? So again, very curious to know what your answer would be.
Yeah, this is always a good first question, isn't it? It's challenging. I hold the view that the brain is physical and our soul exists in an area outside of the brain. There are some neuroscientists that feel that they have found parts of the brain where our consciousness lies, which for some folks is a synonym for the soul. I disagree. From my standpoint, the soul is spiritual and does not exist within the boundaries of the physical brain, but God does allow those to interact. I think there's not a lot of strong evidence to support that claim, but I think this question exists outside of what we can identify. I think some folks have suggested, for example, the pineal gland is a seat of consciousness, and I would disagree with that. So I don't have strong evidence to support that, other than for me, my Christian beliefs come first, and it's not necessary, in my view, for those to be woven together entirely. I hope that makes sense. I'm not trying to cop out here, but I don't believe that there's great evidence that these are woven together and that it's impossible to separate them.
So you, if I understood correctly, you don't see the soul and the physical body interacting?
I think they interact. I think they're not merged together. Like I don't think our soul, for example, exists inside of our brain. But I think when our brain decays and or some of us may have a better brain. I'm not sure that's independent of our soul's activity.
Okay. All right. Thank you. We had yes, right here.
Yeah. So earlier in your talk, you talked about the size of the brain, and its relationship to intelligence. And so I have three questions in relationship to the reptilian and the mammalian brain. One, what's the origin of that concept of reptilian and mammalian? And two, from your perspective, what's the validity of that thought? And three, what's the current science on that?
Yeah, it's a good question. And it depends on the perspective from which you begin. I am a creationist and feel very passionately about the validity of Scripture outside of what is largely considered to be a quintessential evolutionary worldview. So I disagree a lot with colleagues of mine that share beliefs in the evolution of the human brain over time from different species. So I guess I won't comment on that, other than to say I disagree with that perspective, and I think God designed us independently of those efforts. I think there are commonalities between brains across species, and it's easy to, as humans, we seek patterns, as I mentioned, but I don't think, for me, that's not a guiding principle in how I view the brain. So that perhaps answered all three at the same time, and not to shut you down, just that I acknowledge what my field broadly believes about the evolution of species and the commonalities between different brains, and how humans may have the best, but I disagree with that worldview.
Okay, right here. Right here. Thanks for coming, given what's going on in your neck of the woods. I appreciate you coming and being able to get out. Could you comment on the current state of research into Alzheimer's and dementia, and if there's a link back to what you're working on? I'd love to hear it.
Yeah, so it's not, I would say I'm not heavily involved in neurodegenerative diseases, but I'm aware of current therapies that have been approved for these. For the most part, they are an early stage disease, so mild cognitive impairment. The treatments that have been recently approved, I think, have minimal impacts on folks that have moderate to severe disease, and that's unfortunate. So in that sense, they're not disease modifying, but I think they can, for folks that are caught very early on, change the trajectory of treatment. I think what's difficult for these diseases is early identification. So we know that folks that go on to develop Alzheimer's disease show perhaps symptoms of this disease over time, but it is hard to disambiguate or disaggregate that from other features of aging. So I think there's a long ways to go there, but at least there are some therapies that have been recently approved, and those are the first in a very long time. So that's encouraging.
Oh, hi. I'm maybe not going to generalize well from your specifics because I didn't understand them all, but it seems like you separated out learning in adulthood from pre-wired things, but I didn't know, and it's occurring to me just from the news, there are professors who say that reading to our children will interfere with equity because they will do better than other children, so we should stop. And it brings to mind the fact that you didn't talk about learning in childhood, I didn't know, or in use of an animal. Why did you separate that out? What's the difference? And you got saved at age 6. Have you ever speculated on why that seems earlier than the majority report where everybody says get saved at 13?
So maybe I'll answer the last question first before I forget it. My dad's a pastor, and both of my grandfathers, so those I think help to predict an earlier salvation experience. With regards to learning in adulthood, most experiments on mice take place when they're 3 months of age, when they're considered to be adults. So that's kind of the gold standard upon which we can compare these data to previously published data. So that's where the focus on adulthood comes from. And interestingly enough, most experiments are done in male mice. Female mice have hormonal changes that male mice do not. I think that's another, maybe somebody has a question on that topic. That is a limitation of science, biomedical research broadly, that it's all, for the most part, being done on male animals and male humans as well. But those are ways that scientists try to help generalize the findings across groups. I suspect if we did the same experiments in very immature mice, we would find largely the same results. I think our lab at that time had done early investigations there. It is harder to record in younger mice because their skulls are much more flexible and prone to tearing. When you're implanting these electrodes, you have to drill into their skull and put some screws in, and this has to sit on their head over time. You can imagine for something that's already very light and small, it's difficult. So I think the reason to avoid that here was logistical and helping to make fair comparisons with past data. I think now there are ways to do this recording where it's wireless, and the recording devices are much lighter. So it would be interesting, and folks probably will do this, repeat these experiments on mice as they develop. It's possible to delete these receptors a little bit earlier on than we did here. If you delete them, not conditionally, if you delete them and you just have a double knockout of the NMDA receptor, those mice would die. It would not be born without that. So that's why it has to be conditional upon the time of their adulthood.
Okay, I have 2 questions. First one, when I think of a pink elephant, if a neurosurgeon go into my brain, is he able to recognize the electrical pulse or whatever that neuron things that produce that pink elephant?
So that's an interesting. These are good questions, by the way. This is kind of like Jeopardy. Let me back up and say a highlight of what I've shared today is the specificity at which we're recording neuronal activity at the level of an individual neuron. Perhaps you have had an MRI or a similar scan, and that's showing activity patterns broadly in a brain region. You're not getting cell level specificity. So that's a highlight and an advantage of this type of recording technique. Unless someone was able to go in and record on the level of an individual cell in your brain. They would not be able to replicate this. And even further, imagine how many thousands and thousands of cells that you have. For them to find that one, if there's just one, would be tremendously difficult. What can be done is a scan where we're showing you different, did you say a pink elephant? A pink, or whatever. You wanted a pink elephant. So we're showing you a pink elephant. Maybe you have multiple brain regions that light up, but that specificity is not there. A second part to that answer is that we know that there are cells in the brain that are specifically tuned to objects. Like there are people have done experiments. There's a Halle Berry cell, for example. There's a cells in the brain that respond specifically to the concept of a nest. So there are very specific encoding cells in the brain, and that further validates what I'm showing here, which is that we've uncovered these, but there are also ones that respond that are tuned more broadly than that.
So second question is, look like you did the PhD, and many people go into that field, lose their faith. Looks like your faith has increased. Can you tell us more about how your faith increased? Or when I say faith, I mean believe in God, like got strengthened through your field of study. You can elaborate on that.
Yeah. As I mentioned, my passion for science is driven by my love for the Creator. So I met Him first, and that has been a guiding theme in my life. So certainly I understand other arguments that are outside of my faith. I understand the theory of evolution and different narrations of that. I understand the other side well, I think. But for me, the weight of that evidence, if you will, is very light compared to the Bible, which I view and hold with ultimate authority. So every piece of evidence that I found in my research journey has just supported what I know about my faith. And I remember one day when you do these experiments, sometimes you're sitting in a dark room, you're by yourself, you're looking in a microscope. Maybe you've been there. It's kind of lonely. But I was looking in a microscope, and I had asked a question about expression to see if this was expressed there in this way. So I was answering this microscope experiment was answering that question to me, and I saw that. And I saw the answer to the question, and the first thought I had was, "Oh, that's how God did that." That's how God did that. And for me, my science is almost a form of worship in that regard. And the Lord and I had a wonderful time in that microscope room together because I think what I found there further validates my belief. So I hope that answers your question. I understand and acknowledge and appreciate alternative views. And I'm in academia and in the research world, it's a part of it. But I think God has allowed the evidence that I've seen to strengthen that. And it's outside of neuroscience as well. Like John mentioned, I enjoy archaeology, I enjoy paleontology. If you're in a scientific field, I probably like your field a lot. And God has used that in my life.
I think your answer would be similar to a lot of the really great initial thinkers in science who said they were trying to think God's thoughts after him. I'm trying to see that. Yes, so Rich in the back.
Yeah, I've got a question about your research. But first I want to say thank you for the response you just gave. That was really amazing. Okay, on your research, essentially, if I've understood you correctly, which may not be the case, you said you discovered evidence for patterns in neural responses that are not that exist independent of learning. So that raises the question: hey, what's giving rise to those patterns? Now, the genetic analogy would be DNA has the information that produces the configuration of proteins. What are there leading theories about what could account for what you're discovering, or is this because it could be evidence for intelligent design, or not, but maybe yes. And I'm just wondering, what's the state of science in terms of trying to explain what you've discovered?
Yeah, it's an interesting question, and I think both of the explanations that you offered are ones that I hold to. I certainly view this through the lens of a Christian worldview, and this to me is evidence that it is difficult but not impossible, for me to believe that this happened haphazardly over a long period of time and somehow happened in multiple animal species across brain areas, within brain areas. So that's difficult, but not impossible. I'm able to hold that thought at the same time. I do believe that it is largely genetic. What's interesting about it is that I believe God enabled flexibility through learning and through the aging and developmental process. So I think what we find often in nature is that genetics set you up, and then these are modified over time through development, through your environment. So I think the answer to me is both, and I don't think most scientists would disagree with this being with a genetic component of what you shared.
Me? So this has to do with the appetite testing in the mice. Did you see variation between mice with preference for, say, the mice food versus the sugar versus the rice versus the milk drops? Because I know for me, I can smell raw onion across the room. I hate raw onions, type of thing. So did you see that kind of appetite variation?
This is a good question, and this was one that would arise in a conference. So I'm glad you asked this. To avoid that, mice have their food limited for some time before the experiment, so that there is a hunger drive. And in that case, when there is a hunger drive, there's no real preference. It's kind of like, if you're very hungry, yes, I'll have Chick-fil-A or I'll eat McDonald's. But if I'm not hungry at all, I definitely want In-N-Out. We've all been there before at the airport. So there was no preference in this study. And another reason why I can say that is the case is that we presented them individually. So it will be one rice grain, and they come up to it. You analyze the events at that time, and then one rice grain. So they're never presented the option of all four at the same time. So that prevents kind of the preference and any changes related to coding that would happen for that reason. But it's a different, interesting set of experiments. Yeah.
Dr. Cox, my name is David. Thank you for your presentation and bringing it to a level, a very complex topic that I think I understood the piece of it. I had two questions, and I think maybe you answered the first question at the conclusion of your presentation. You said something on the order of, I think there's some relationship to artificial intelligence, the developments that's going on in that space. And I don't really have any more questions there. I saw a parallel throughout your presentation. The second question I have is more developmental oriented in the fact that there is some developing in our world today, and I'm thinking of the Neuralink work that Elon Musk company is doing, where Neuralink implants are being already approved by the FDA. Can you comment on that, because it seems so closely related to your topic that today someone can think about activating an arm, and it's a mechanical arm, and it's activated. So that Neuralink topic is my question. Are we there already?
Sure. Is anybody here from Tesla? I know this is a Tesla. Just confirming. Okay, I don't think Elon's here either. It's too bad. I've seen a lot of them. I actually, yesterday I saw a fire truck that was a Cybertruck, and I thought, wow, these people in Austin are rich. I don't know if that's the case. I don't know what happened there. We can talk about it later. But it's a great point. And actually, when Elon was first doing those experiments in pigs several years ago, it kind of upset the scientific community, because folks like my team and others have been doing that type of research for a very long time with greater accuracy and stability. But as he does, he is able to sensationalize things. So he repeated what has been done well for a very long time. In terms of what's being done in humans, that is cutting edge, and that kind of addresses the question of the gentleman over here, that we have not, up until very recently, had cell-specific specificity in recording responses. It's been, oh, this part of your brain lights up when you see a cheeseburger, this lights up when you see a red truck. now there is a possibility to not only uncover those patterns, but as you mentioned, modulate them. So first, if we understand what's happening, how can we modulate those patterns? It's similar here that an obvious next step for this type of research is to ask, how are these coding patterns changed in different disease states? How is it changed in Alzheimer's disease models or Parkinson's disease models or schizophrenia? Are there ways for us to, because you know the drug targets for most of these diseases are not unspecific, are non-specific to what we're talking about here. Perhaps this is another angle through which to get at those diseases, which up until now have been very hard to target. So I think it's absolutely the right next step and an extension of work that folks have been doing for a long time.
Amazing.
Thank you. Question?
I somewhat understand the idea of the neural network that there's this crazy interaction of all these neurons. Is there any understanding of how this becomes a unified perception? Like I'm looking at you and it's made up, like all my cones and rods are putting together different signals, yet I see a unified picture. There's also sound that's unified with the visual, could be smell and touch. I kind of see how many comes into maybe a clique, so to speak, of a group of cells, but how does that perceived as one unified perception?
Yeah, that's a great question. And you sound like a grant reviewer that I've received feedback before, so that's an interesting question. You can imagine that the research that I presented here is the very first attempt to get at testing these principles. Another obvious next step would be on top of all these, is there a meta-unifying principle? And I think the meta-unifying principle takes convergent inputs from all of the cell assemblies that we're talking about. So your perception of me is auditory, it's visual, it's somatic. You're bringing in your own conceptual knowledge from past events. I believe that what you're talking about lies on top of those, and that's kind of those general cells converging upon one another to make a very abstract concept. So I do believe that something sits on top of what we're talking about here at an individual level, and that it's convergent in nature. But that's something we've not tested. I think it would be very difficult to do so in mice because they lack language skills and things that would allow us to get at those great details. But I think down the road in humans, perhaps that's something that we can explore further, where we're able to get data other than the spike rasters.
Okay, one more question, Chris.
I have a question about your research in the receptor knockout mice. How do you know that they are not capable of learning and memory?
Yep.
Did you verify that in the experiment, or are you just going off of past knowledge?
Yeah, so my thesis advisor published back-to-back papers in 1996 in Cell, specifically focused on these mice, and showed that they have impaired learning, memory in the short term and also long term. So we can go back to those experiments and show that is the case for these mice. And to convince yourself, we also repeat these experiments. When we do these individual new ones as well, so you can go back to the past literature, which we've shown. I think that in the days where I was not reading, he did this research, so we can always go back to that. And it's also these mice are grown and kept over many years in laboratory settings. It's important to do sense checks of this was the state in 1996. Is this still true today? And the answer is, generally yes, but it's a good sense check.
So I'd like to close with this last question. So in high-level talks like this, one of the things that I hope that everyone is able to take home is the gist of it. And so to close with trying to summarize the gist of it, in your description of the talk, the phrase that really stuck out was pre-wired for connectivity, and how that could be interpreted as evidence for creativity. If you could just sort of summarize in layman's terms how this research has done that. I think that'd be a good way to close.
Yeah, absolutely. So we all have brains that are filled with thousands of neurons that are connected with tens of thousands of other neurons, yet we need to make sense of what's happening today, what happened yesterday, and what happened tomorrow out of what's a milieu of what could be random, happenstance. I believe that we've shown evidence that there is a pre-wired, pre-designed computational motif that is embedded in all of our brains, regardless of whether we realize it or not, that indicates that there was an intelligent design behind each of us. And my prayer is that this evidence will strengthen your faith. And if you have no faith, I think this is a great place to talk about it with others. And that's my prayer, that this evidence can be used in your own life. I realize you're not all going to go out on Monday and start a lab and doing surgery on mice and scaring them like I have. But I feel that this applies broadly. You can be confident in knowing that God has created you for a special purpose and has given you a mind that is able to fulfill that in any way that He sees fit. So thank you all. Really appreciate it.